Good afternoon, everyone, and thank you all for being here today. My name is Echa Osara. I'm the project manager of the Health Resources and Services Administration grant with the Southeast Regional Genetics Network. The grant PIs are Dr. Rani Singh here at Emory University and Dr. Hans Anderson, who will be joining us via WebEx from Tulane University. As part of the grant initiative, Emory University hosts a few lunch and learn events throughout the year with the goal of highlighting key experts and their research within human genetics. Before we get started and before I present the presenter today, I just have a few housekeeping items. We have our parking vouchers at the back of the room, so please grab one before you leave. There's also a pre and post test that we're administering, so please complete those as well. And there's a sign-in sheet going around also. Our speaker today is Dr. Carla Cuthbert. Dr. Cuthbert is the chief of the Newborn Screening and Molecular Biology branch at the CDC. And today she'll be giving a presentation on the lab detection of patients with GAMT deficiency. Please help me welcome Dr. Cuthbert.
Thank you. Thank you for inviting me. Again, my name is Carla Cuthbert, and really happy to be here. I think this is the second time that I've been back to Emory to present, and I'm just going to be talking a little about GAMT deficiency. I'm going to give you a little bit of background about it if you're not familiar with the enzymatic pathway. I'm going to talk a little about how it's detected within the laboratory. I'm not going to go into elaborate amounts of detail. Like if you are able to bring up amino acids and acylcarnitines in your laboratory, it's fairly easy to add on. I will be also finishing up with some of the information that I presented recently in Washington, D.C. About newborn screening for GAMT deficiency and that status worldwide. Is that a little better?
Yeah, thank you.
Okay, so creatine. I really like to see structures, so I had to throw in that structure for creatine. It's a non-essential dietary amino acid derivative. It is synthesized and stored in in different locations within your body. So it's synthesized within the liver, and it's stored in areas where there is the greatest high energy demand. And so over 90 to 95% of your total body creatine would be stored in your skeletal muscle, and your brain is also one of the locations for creatine storage. So creatine is really important in that it provides a critical energy source for your body. ATP and ADP can only get around so far. Creatine stores when it gets converted into creatine, the phosphocreatine. It has a high energy phosphate group that it can use to replenish ADP and convert ADP back into ATP within places within your body. So that's a very important function. So it works very much in concert with ATP and ensuring that you have enough energy storage within your muscles and within the brain in your body. So as far as the creatine synthetic pathway goes, this picture just shows, again in red, the pathway that will eventually lead to a creatine synthesis. Sorry, do I have a pointer here? Yes, okay. So here we start with glycine and arginine. The two enzymatic steps involved in the process is through the enzyme called AGAT, or the L-arginine glycine amidinotransferase. And that transfers an amidino group from arginine onto glycine. And that forms the guanidinoacetate that we see right here. The guanidinoacetate gets a methyl group transferred to it from acetyldenyl methionine, and that happens through the action of GAMT itself to actually form creatine. Creatine can spontaneously convert to creatinine, which I think many of you would be very familiar with, and that gets excreted at a very constant rate every day. So it's very important to ensure that you have replenishment of your creatine pools. And in terms of maintaining your body creatine pool, creatine comes from both the synthetic pathway that I just talked about, as well as dietary uptake from things like fish and meat. So those are very important sources of creatine to ensure that your body has all of the creatine and energy that it actually needs. So this pathway is very important, together with food intake. Once creatine gets into your blood, it's taken up into your tissues by the creatine transporter, and that allows for distribution of your creatine within your body. Creatine at these points within your body can then be phosphorylated to phosphocreatine and serve as your energy reserve. Again, creatine can be spontaneously converted to creatinine as well. In terms of the deficiencies associated with with creatine biochemistry, there are really only three. AGAT deficiency is an autosomal recessive disorder. The enzyme is located specifically in the pancreas, liver, and kidney. And patients with this disorder have a very nonspecific clinical presentation that includes developmental delay, marked speech disabilities, autistic behavior, hypotonia, and failure to thrive. Patients with GAMT deficiency will show an autosomal recessive pattern of inheritance. This disease does show that. The tissue expression is very similar with AGAT, and the enzyme activity can be found in pancreas, kidney, and liver. Patients with this disease show onset typically within the first couple of months to the first couple of years, and speech delay is very pronounced, together with dysfunction of tone, developmental delay, and movement disorders. Of note, about 90% of the patients will have seizures, and movement disorders and other severe presentations are common. In adults who have this disease, they show autism with self-injurious behavior. Creatine transporter deficiency is X-linked, and it's expressed in most tissues, as you would expect, because you expect to have creatine being picked up by more tissue types. Children with this disorder have mild intellectual disabilities, seizures, hypotonia, again, more severe language delay and speech disorders, and slowly progressing disorders. Adults have static encephalopathy. They've indicated that this disorder has been identified in 1 to 3% of patients with non-syndromic X-linked intellectual disability. So in terms of disease prevalence, this is something that will come to bear later on in my talk. Patients with AGAT deficiency, less than 20 patients have been identified worldwide. GAMT deficiency, which is the second most of this particular group, only about 110 patients worldwide have been identified or reported. And the creatine transporter, they have a little bit more, just over 150 patients have been identified. It's been said that these patients are likely underdiagnosed since they have very nonspecific clinical phenotypes, and you really need to think about it to be able to either do an MRS or do the biochemical. They've indicated that it's one of the more important metabolic causes of intellectual impairment and autistic behavior, and should really be considered when you're seeing these patients. So in terms of treatment principles, patients with AGAT deficiency just really need to have creatine supplementation. Patients with GAMT deficiency get creatine, so creatine supplementation is helpful. Arginine restriction is also very helpful here. Ornithine supplementation is helpful, and benzoate treatment is helpful to mop up the excess glycine, which will be excreted as a hippurate. For patients with creatine transporter deficiency, haven't found that they've indicated that creatine supplementation may help, but may not always be effective. So in terms of the laboratory detection of patients with GAMT deficiency, and I'm going to focus on GAMT just because that's the main title of what I'm speaking about. I just want to refer to this, this reference in Genetics and Medicine. Dan Scherer, many of you may know him, together with a number of metabolic specialists across the country, have put out a really good paper on the laboratory diagnosis of creatine deficiency syndromes, and this you should actually just pay attention to if you're planning on bringing on any of these methods. So I just wanted to highlight that the proton magnetic resonance spectroscopy is really one good way of being able to detect deficiency of creatine, of cerebral creatine. And I just have at the left here, patient without therapy has an elevation of GAA with a decreased amount of creatine over here. With arginine treatment, we see that the creatine still hasn't gone up a whole lot, but once you start supplementing with creatine, that does get elevated. One of the concerns, again, with this is that it's expensive and requires sedation or anesthesia in children. So the initial diagnosis is typically established by measuring some of the key biomarker elements in plasma, urine, or CSF by using LC-MS and looking at creatine, guanidinoacetate, and creatinine. So if you are looking at a patient who is presenting clinically, you may want to determine whether or not what the metabolite patterns for each of these disorders are, and I've just shown this in this particular table. The one thing I want to highlight is that with GAMT deficiency, you're going to have an elevation of guanidinoacetate, and you're going to have low amounts of creatine. Again, these are two enzymes that are involved with creatine synthesis, so you expect that if either one of them is deficient, you're going to have a reduction in the amount of creatine within your blood. So elevation of GAA for GAMT deficiency and reduced amounts of GAA for AGAT. So LC-MS is typically used by using stable isotope dilution mass spectrometry, and this generally gives very good sensitivity and specificity. In an under-derivatized, you can set up your assay either in an under-derivatized or a derivatized form, and it's very, very similar in terms of the approach. And again, if you set up your amino acids, acylcarnitine methods, you would be fairly familiar with this particular process. Essentially, you add stable isotope standards to your samples. If it's plasma, you need to deprotonize and clean up your specimen by centrifugation or filtration. Then you concentrate your samples by drying under nitrogen and resuspend in an appropriate solvent. And that's if you're not derivatizing. If you're derivatizing, you do several of those steps, but you reconstitute with butanolic HCl, incubate for about 15 minutes, then dry under nitrogen, reconstitute, and you can run your samples. Again, you would have to do your method development. You'd have to optimize your conditions on your mass spec to make sure that you can actually detect the analyte with the right sensitivity and specificity. And of course, you would have to do appropriate validation to make sure that you can detect the marker in patients who actually have that disease. So those are all things that are expected whenever you're bringing up a specific assay like that. For GC or gas chromatography, if you're doing the electron impact method, it's a two-step derivatization procedure that is followed by analysis on a nonpolar column. You incubate with this particular compound to stabilize the guanidine group, and then derivatize and the product is then run on your GC. With negative chemical ionization, it's also a two-step derivatization process, and these are run on polar columns with your GC method as well. Again, it really depends on what you have set up and what you're planning on doing, and your throughput and what your expectation is for running this particular assay. So verification of a patient who has this particular disease is generally done by enzymatic, by looking at your enzyme activity or by DNA evaluation. You can perform an assay for GAMT activity in fibroblasts, or lymphoblasts, or amniocytes. The substrate would be a radioactive or stable isotope labeled guanidinoacetate, as you see here, because we're focusing on the GAMT activity. And you're going to be measuring the increase in labeled creatine as a result. And you would have your activity be a function of the concentration of creatine that gets formed. Okay, so of course, like I said, method development, validation, all of these things are necessary to establish your appropriate reference ranges for normal and affected samples, and these are very important, necessary steps that you would do as you're setting up your assay. For molecular testing, the gene just spans about 5 kilobases and consists of 6 exons. So you would do this by Sanger sequencing, ensuring that you sequence the exons, sequence the intron-exon batteries, the promoter region, to determine whether or not the variants that you identify have any significance or not. So the programs that are currently doing testing for GAMT deficiency are following up with sequencing analysis, just because they want to know and build a repository of variants that are associated with this particular gene. So in the last couple of weeks, I indicated that I gave a bit of a presentation at the SAC-DNC in Washington, D.C. It had been 2 years since GAMT deficiency had been nominated for addition to the recommended uniform screening panel. And I don't know how many of you have been at Emory that you remember Nicola Longo and Marzia Pasquale, but they have a very strong Emory connection, and they're currently in Utah and were the nominators for this particular condition. And just so that you understand that there is a very significant process for being able to nominate and to get a condition onto the RUSP, I just really quickly outlined it here on the bottom. We have a nominator in the community who submits a package for nomination, and there are specific things that they need to identify. That package gets reviewed by a separate work group. It is very expensive to go through the evidence review, and we want to make sure that we have enough evidence for the evidence review committee to actually come up with proper evidence. So it was reviewed by the nomination and prioritization work group, and then presented to the SAC-DNC committee for a vote. And it was upon this particular vote at this particular time that it was determined that there was not enough evidence for it to actually move on for evidence review. And this was in 2016, it's now 2018, and Dr. Boccini, who is the chair of the SAC-DNC, wanted an update about where the states actually were, where the programs worldwide were, and whether or not anybody had identified any new condition, any new cases. So the next couple of slides will just identify, would just be some of what I presented to the committee. And this is an excerpt of the letter from Dr. Boccini to Dr. Longo about a year ago, about 2 years ago. And essentially he was saying that at this time the committee had decided that the evidence available did not meet the criteria, and the committee decided not to move forward with the nomination. Okay, so they were encouraged to continue to resubmit as more information was given. And just in terms of just summarizing the deliberations in 2016, the things that were in favor of adding GAMT deficiency to the RUSP were here, and those that worked against it are identified here. The natural history has been understood. Treatment is very similar to many of the classic inborn errors of metabolism. The outcomes are best when there is early treatment. Newborn screening assays can be multiplexed with existing tests. That should be laboratory-developed tests specifically. And there's a newborn screening. There's a newborn screening strategy that can give us high sensitivity and specificity with low false positive rates. The understanding of this disease, however, was just based on 110 reports worldwide. And at this point, there was no agreed upon treatment strategy, although in 2014 there was a really good paper that described the treatment approaches from several patients with GAMT deficiency. So we found that this was less of a concern when the metabolic physicians pretty much agreed we can handle these patients. Metabolic control, of course, must be strict. There is no FDA-approved newborn screening or diagnostic assay. And the biggest issue is that no patient had been identified through the newborn screening program. So Dr. Boccini indicated at that time that we would not initiate an external review because no case had been identified prospectively through the newborn screening programs to date. And that would hamper all of the metrics that we would be looking to determine. Treatment guidelines should be addressed. So they sent the proponents to work on some of these issues, and I've been in contact with many of these laboratories to find out whether or not they've had cases, and I will just indicate to you some of the summaries that we've had so far. So the newborn screening program that has had the longest and the greatest amount of newborn screened to date is in the state of Victoria in Australia. And that particular population, they have about 70,000 births per year, and they began screening in 2002. And Dr. Pitt, when he set up his laboratory, he's very good at mass spectrometry, and so he's also a biochemical geneticist. So he thought, what can I put on this that would make it worthwhile? And he just put together a panel of analytes that he thought would be relevant, and he included guanidinoacetate on that fortuitously. So he did that. He's screened at several, done some repeat testing on some that have come back with an elevated guanidinoacetate, followed up some of these cases, but they were all found to be false positives. So again, found to be false positives through assessment of urine levels of GAA, creatine, and creatinine, and they were shown to be non-consistent. Sorry. One of the things that Dr. Pitt indicated was that if he were to now present this sort of data to his committee with a number of cases that have been screened, and he's screened so far for about just over 1 million babies in that length of time, it would be really hard to get any kind of support to add GAMT deficiency on the list at this point. So it was very fortuitous that he just included it. It's going on in the background, and it's continuing to be screened in the background. Again, they're not aware of any false negatives at all. They have a setup in which they've got a newborn screening laboratory, and just across the corridor, they've also got the biochemical genetics laboratory. So if any case had been clinically ascertained, he would actually know about it because he runs both of those labs. So there have not been any false negatives to date. So it seems that it's very rare in this particular population. He has no intention of stopping screening, so this will continue to go on in the background. I keep pressing that button, I'm sorry. Our colleagues in British Columbia and Canada decided that they would also set up this assay as well. And they did an initial pilot that started in September of 2012, and they ran their pilot for 3 years, screening all of the infants in British Columbia. And currently, it's running again in the background as a routine test. And Graham was kind enough to share his screening algorithm with me, and just wanted to be able to share that with. I just wanted to be able to share that with you. So again, a newborn screening card is punched. All of the samples and all of the assays are run through that first tier test. This is very similar to the amino acids. So it's this is a single punch in which you do amino acids, acylcarnitines, and then you also have the guanidinoacetate in that particular assay. And if there is a screen positive, so if there's a GAA over 3.5 micromolar, that gets run as with a second tier test. And what they've done here, this is has liquid chromatography attached to it so that there's better separation. And they've integrated this particular marker, GAA, with the MSUD panel as a second tier assay. So they're actually able to run this as well. So if that is also positive, then they will just take it to sequencing to look for variants and determine whether or not they have a positive or negative screen. Then they'll follow up with calling that newborn in and looking at urine and other specimens to verify. Okay. And in British Columbia as well, the number of newborns is approaching 250,000. And to date, they have still not found any true positive cases. They've identified 2 screen positives that were followed up, as I said, with GAA and creatine testing in plasma and urine, and both of these tests were found to be normal in both cases. So those were false positives. So again, they're not aware of any false negative cases. BC is small enough. And they're collaborative enough that they would know if there's any cases that they might have missed. So again, in this particular population, the incidence is still not known. And in the state of Utah, Marcia and Nicola are commandeering screening for GAMT deficiency. It is currently a routine screen, and they've been screening since February of 2018, and they're approaching about 140,000 newborns having been screened. They've had 2 false positives, no false negatives. These again were found to be false positives when you tested urine and plasma. Again, no true positive cases identified. The state of Michigan is also intent on bringing this on board, and I think Mary Cederling is listening in as well. So we're very excited for other states to be bringing this on. CDC is ready to help any of the programs that are willing to make modifications to their assay to actually do this. It's interesting because when I speak to the moms who are very strong advocates for GAMT deficiency being added to the newborn screening panel, when I put on my biochemical genetics hat, it's like, this would be such a great thing to have added to the panel. You know, if there's any state that wants to do this, we're there with them. We have all of our methods ready to go. We have materials ready for them. But when I put on my Fed hat, when I have to sit at the table to make a decision, we have to comply with what the criteria is. And one of the big criteria is that we need to actually have at least one case pre-symptomatically identified through newborn screening, and that to date has not happened. So to date, CDC is still unable to say yes to the addition of this condition. But we're very excited with Michigan, and we are looking forward to being able to work with them to help them bring on this particular assay. So as I indicated, CDC we've published on a method using non-derivatized assay for combining guanidinoacetate with acylcarnitines and amino acids. There was already a published paper using the derivatized method. So we have both approaches available. Depends on what the states or any other program is interested in doing. We'd be willing to help. And certainly we can help states with method development, validation, and implementation at any part of their testing, which includes just review of their data, troubleshooting, conference calls, on-site visits, hands-on training, anything that they need, we'd be willing to help them with. We have quality assurance materials available, especially this is very important, especially when it's such a rare disease. You really want to be able to have the right concentrations of creatine, creatinine, and the guanidinoacetate. So we have that already made. And starting in 2019, we're going to integrate the guanidinoacetate in together with the routine amino acids reference material. So anybody who already has it as part of a combined test, where they're looking at guanidinoacetate plus the amino acids and acylcarnitines, they'll be able to detect it. So those who are not screening for it, they won't see it. So it'll be blind to them. So that's something we hope will be helpful. And again, we're happy to support and help any state that is planning on bringing this test on board. So that's all I've got for GAMT deficiency. Of course, it's a very rare condition, but it has a serious medical outcome, and it can be detected very well by LC-MS with an elevated pattern of guanidinoacetate with reduced concentrations of creatine. The treatment is very similar to some of the other conditions, metabolic conditions. We supplement with creatine, ornithine, and benzoic acid, and restrict arginine. To date, approximately 1.4 million newborns have been screened for GAMT deficiency in 3 programs across the world, in Victoria, Australia, British Columbia and Canada, and Utah and USA. And none of these programs have been able to identify a case pre-symptomatically. And of course, CDC is ready to help any program technically or with materials so that they can bring this condition on. Okay, so how about that post-test? Do you want to, can I give you like 2 seconds to take a look at it? And we'll see, we'll go through it. We'll go through it really quickly just to make sure that you get the answers correct. So I'll tackle the first 3 questions, and you can spend more time to answer questions 4 and 5. But the first question was, which one of these statements was not true? So the first was GAMT deficiency has an autosomal recessive pattern of inheritance. That's true. Over 90% of the creatine pool in the body normally comes from dietary sources. That is false. 50%. Creatine is phosphorylated to phosphocreatine and serves as an energy reserve for ATP regeneration. That's true. Patients with GAMT deficiency can present with speech delay, seizures, and movement disorders. That is true. And patients with GAMT deficiency can benefit from supplementation with creatine, ornithine, and benzoate, and restriction of arginine, which is also true. Number 2, untreated patients with GAMT deficiency will have which plasma biomarker profile? I'm not going to read them all out. I will just say that the answer is C. Yeah, C. So it's high guanidinoacetate and low creatine because it's a creatine synthesis enzyme. Which statement is true about newborn screening for GAMT deficiency? The first, approximately 1.4 million newborns have been screened for GAMT deficiency in newborn screening programs worldwide. Yes, I just said that, so that's true. No newborn with a confirmed GAMT deficiency has been identified through newborn screening to date. That is also true. Newborn screening for GAMT deficiency was added to the Newborn Screening Recommended Uniform Screening Panel in 2016. That is not true. So the correct answer is E. Statements A and B are true. Okay. Any questions at all?
Thank you so much. That's very insightful because I keep hearing about the GAMT deficiency and there's some concept that it's going to be added soon. So I feel it was important for us to bring this to our region, the truth, what you enlightened us with. I think as a nutritionist sitting in the audience, the data I saw on treatment, with arginine or low protein diet plus that, it's very mixed at this point, the outcome, even though there is a treatment. But I think it was implicated that all the patients, if I'm correct, all patients have been picked up clinically. Are there any patients, to your knowledge, picked up by newborn screening and treated early on where we know the outcome was really good on this treatment?
To date, there is no patient that has been picked up by newborn screening prospectively like this and then treated. That is the one thing that we're waiting for. That's the one thing that I think that's everybody is waiting for. So to be able to find one case, and even when we were discussing the requirements for pilot studies and so on, one of the questions was, well, is one case enough? Because how representative is one of your patients for any group of diseases that you're actually looking at? So, so that becomes a bit of a concern as well. I think that we would certainly have another discussion for consideration when we get that first case, but we have not found that patient yet. So we can't answer those questions about treatment at all.
If nobody has other question, I'm going to ask one more question. So I happen to be sitting next to a mom from Utah who was picked up by Nicola, who's in the advocacy board. And the dilemma you see with the mothers who feel their kid could have done so much better. And it's the incidence, at least in the literature from the European, it's very rare, right? And so you need a state with big births. Utah, it can take years just to reach. The birth rate is, like in Georgia, I think it's 120,000 or 130. And but what about like Texas and California? Have they stepped up to volunteer to do something like this with CDC or to see the numbers so we can get some like bigger? I don't know the Michigan's birth rate, but to me it's a story. We may have to wait 2 or 3 years for the first case if we are going after small states. Is there any activity from Texas, California, Florida?
Those are all good questions, and I think that any program that is thinking of trying to amass a certain amount of data to get enough evidence for being able to present to the committee wants to be able to target a state that has a high birth rate. To my knowledge, none of these states are planning on bringing something on. From my point of view, from the way I see things, and certainly this is not me, CDC, thinking like this, but this is just me based on what I see. I think that if a state is thinking about making a change to their amino acid or acylcarnitine assay, I would think that they may consider adding something on, because one of the challenges, again, that any state will have to go through, if they have a laboratory-developed test, they make a change, they would have to revalidate that particular assay again. They'd have to make sure that it's still running the same way that it was, and with the new change, it continues to function well. Perhaps if a state is already making a change for another condition and it wants to add this on, that might be something that they may actually do. But with Michigan, they've decided that they would just do it. They had lots of conversations with their advisory group, and they made a decision to actually add this condition to the RUSP. So it's not something that is done very lightly. They do have to consider the metabolic physicians who will have to be seeing these cases. But from what I understand, the number of screen positives that they identify is very low. So that's why there is a sense that the sensitivity and specificity is fairly good with this assay. When you're using, again, a first tier followed by a second tier with liquid chromatography, and if you can get some sequencing in there as well, that would also be helpful. So, yes.
At what age have the individuals that have been identified, at what age have they been identified? Because newborn is not capturing it, so at what age are they being identified as having this disease?
So again, it depends on the actual reports, but usually these patients are clinically ascertained. So many of these patients will be presenting with things like seizure, failure to thrive, developmental delay, speech disability. And so again, it really does require the attending physician to think about doing the MRS, seeing a reduced amount of creatine, and then following up with mass spectrometry to look for the panel of analytes. So, I mean, ideally, again, with my biochemical genetics hat on, it would be fantastic to just screen the entire population for this condition. But again, we have to abide by the requirements set out for addition of conditions to the RUSP. We need to see at least one case before we tell the entire country to start screening for this condition.
I know Emily's on the line. Emily, I heard something about Georgia. Was Georgia considering or has a research grant, or are they considering doing this?
It has been nominated to our panel, so we're undergoing the review process for the Georgia panel right now. I haven't heard of any research going on in our state, but yeah, so that's where we are with it has been nominated.
So one of the caveats right now, as we're waiting for resolution in the common rule, is that if you are receiving federal funds to do a research project, you have to get informed consent. So that's one of the downsides of people asking me, hey, why don't you fund this? Well, I'd like to, maybe, if I had the money, but this would be considered a research project, and then you would actually have to get informed consent. So, with what Michigan has done, it's fantastic. They've just incorporated it. They found that there is enough evidence out here to make a move on this, and they made a decision internally to move forward as far as routine testing, together with some of the other programs. And so it becomes a very different question than if you're asking for research. But we're hoping that things will resolve itself come January next year.
So, one of the things at the committee I was asking, other than CDC, NIH had funded 3 grants with, I know from North Carolina, I'm blanking out on the name of the physician who's running the newborn screening. Huh? Not Don Bailey. She has the NIH funding to do the molecular testing for newborn screening. So I know there were 3 grants funded: one to California, one in California, and one in Boston, and one in North Carolina. Cindy Powell. Okay. So I was thinking maybe we should have a conversation with her on the molecular level. Again, the birth rate is way smaller. Have they picked up any of the 3, AGAT, GAMT, and creatine? What is it that has happened with their data, whether they were even looking for those or not, or if they could go back and they have the blood spots to look at that gene? That may be a conversation also worth.
Yeah, so many of these programs, when they started, they did a retrospective analysis, so they may have collected tens of thousands of their previous blood spots and done a screen to see what their normal range is, and that's something that is included. Now, prospective means that you have your assay validated, and then you are prospectively looking for your patients. And so that's where it becomes a bit of a challenge. In hindsight, you can tell a lot of things, but you want to be able to make sure that you can identify that case, be able to have an intervention that is timely and that's appropriate and that shows benefit to that particular child. And that's where the big conversation has developed. Some people say we know enough. Others, we have a criteria and we have a list. We need to make sure that we stick to the list before we require all states to actually do this.
I have to tell the story to the audience. This mother who has a child with GAMT deficiency and is really advocating, she, in 2016, when they announced, she was totally crushed because the literature is strong for treatment, but we don't have a case at this time. And she said she's going to go back and get pregnant. And I haven't heard from her, so I'm assuming even if she got pregnant, she probably ended up having a normal child, because she was saying if her child, and she's from one of the states who was testing for it, and said she was so frustrated. She said she's going to go back and get pregnant and see if she has a child to prove that. But so
she would need to be in a state that's actually screening for the test. Okay, good. She's in Utah. Yeah, because yeah, no. She has to be. Okay, because that is also an issue. It's one thing to cast a wide net across the country and then say, well, let's see if we can figure out if there are any high-risk pregnant moms who are going to have this particular disease, and maybe that is something that we could think about. I've been approached to brainstorm about what could be done to be able to be able to find a positive case, and short of trying to contact every clinic that may have patients with these that have these risk factors, developmental delay in speech disorders, oh my goodness, and then have all of them be tested, see whether or not they have moms who can be pregnant, and then track all of those pregnant moms, make sure that their blood spots get to a program that is screening, and then have those, have their respective physicians follow them and track them appropriately. That's hard. That's incredibly hard. And one of the challenges would be that if we do that for this one case, then what would stop us from doing it from another case? But also, you want to test the newborn screening system itself. So you want that mom to be in that particular state, so that you can see how the process can bear up and can support detection and support of that child. So it is very, very challenging, and I wish I had a lot more money. This isn't being recorded. It is. But again, I mean, if I'm kidding. But these are really worthwhile questions, and these are things that it would be really interesting to be able to get an answer for. So.
Are there any questions from other WebEx participants?
Thank you, everyone, for coming. It's really great to have an audience and bring in new topics. Please encourage your colleagues to join us as we bring this. We will have this available on our website. So any fellows or students that rotate through our program can benefit from these educational opportunities. Please share the website, our SERN website, and for the future thing. And thank you again. Please give a big hand to our speaker today.